Okay. Um, so in this session, I will be talking about germline copy number variation. So some of the concepts will be similar to copy number variation in tumors, so somatic copy number variation. But the process for handling uh, germline copy number variant calls is a little bit different. Um, and it will also be good setup for when I talk to you tomorrow about germline structural variant calling. Um, so, and I also want to mention that the pipeline that we used for calling germline copy number variation, we call it GCNV. Um, and it's not a single tool, it's a whole process that you have to run, a whole pipeline. So we'll talk about some of the background, some of the biology underlying copy number variation, how do we model and discover copy number variants, um, and then how do you actually run this pipeline on your own data? Okay, so a little bit of review. What is copy number variation? So this figure, we can see that the segment that's in brown um, appears a different number of times in different people. Um, so that's an example of copy number variation. And copy number variants, or CNVs, are a type of structural variation, a type of SV. So there are other types of structural variants, but structural variants include copy number variants. And CNVs are duplications or deletions of a region of the genome. And you might ask, so we also have indels, small insertions and deletions. So where's the line between an indel and a CNV? And the answer is it's a little bit arbitrary. They're both some sort of deletion or duplication. So where do we draw a line? What we're calling an indel or a copy number variant? It actually somewhat depends on the application of what you're trying to do, and also on the data that you're working with. So, for example, up here, this says CNVs, let's say they're greater than 1 kilobase. That's because a lot of this presentation is about whole exome data, and you get lower resolution for CNV detection in whole exomes. So when you're talking about CNVs in whole exomes, then actually you're talking about larger CNVs, maybe greater than 1 kb. But tomorrow, when I'll talk more about SVs in whole genomes, I might say it's anything greater than 50 bases. So this line is pretty arbitrary. And de novo copy number variants are pretty infrequent. We expect far less than one de novo CNV per individual, but they're a little bit more common at some particular regions of the genome that are recurrent CNV loci. And we'll talk a little bit more about that on the very next slide. But even though there are typically fewer copy number variants per individual than SNPs and indels, they're very large, so they have a big impact when they do occur. And so CNVs seem to be at least as important as short variants in determining the differences between individuals and in playing a role in our evolution. Okay, so I briefly mentioned recurrent CNV loci. So it turns out that many structural changes tend to happen over and over again in certain regions. And these tend to be repetitive regions. So in particular, low copy repeats or segmental duplications, where a portion of the reference genome has been copied and occurs multiple times, as well as shorter repetitive sequence shorter repetitive sequences called microhomologies. So wherever there is repetitive sequence, you're more likely to get CNVs happening again and again. And why is that? The answer is that this repetitive sequence tends to interfere with homologous recombination. So thinking back to our early biology classes, homologous recombination is a normal process that occurs in cells. It happens during meiosis, but it also happens to repair double-stranded breaks in DNA. So if there's any sort of damage to your DNA, maybe from sun exposure even, then homologous recombination is a process that tries to repair that damage, repair that double-stranded break. And it usually does a pretty good job. And this, I'm not going to go over this entire diagram, but it can occur in two different ways depending on whether you have both sides of the break or not. And it's basically using, if you have a break on one chromosome, it uses the other copy of that same chromosome as a template to fix the sequence that that got broken or damaged. So it finds the same spot on the other copy of that chromosome, and it uses it as a template to repair the damage. But if you have a double-stranded break around a repetitive region that is not very unique, then when you go to find that same spot on the other chromosome, you might end up finding something that looks similar, but it's not actually the same location on the other chromosome. And that's when we get copy number variants. And that is through a process called non-allelic homologous recombination. So you're doing homologous recombination, but you didn't find the same allele to use as your template. So if we look at the left side of this figure, we can see that this is a stretch of DNA in which there are two segments that are labeled with an X. So both of those X's are parts of the DNA that are very similar to each other. And if there's a double-stranded break near one of those X's, it goes to find the other copy of the chromosome to use as a template, but it actually finds the wrong X. And so when we go through the homologous recombination process using that as a template, you actually end up with copy number changes. So the output is you duplicated that X and Y on one copy, and you actually deleted an X and a Y on the other copy. So this is all to show you how these repetitive regions of DNA can give rise to recurrent CNVs. This is a process by which CNVs can occur. Okay, so we've just been talking about somatic CNVs and how in tumors, they can accumulate really wild and crazy mutations, really huge stretches of the genome can get duplicated or deleted. And in a tumor, there can be multiple different lineages of tumor cells that all have their own mutations. So you get this whole mixture of genetic mutations. So in contrast, germline CNVs originate in germ cells. So either you inherited those copy number variants from your parents, or it occurred de novo in a germ cell. And that means that instead of just being localized to a tumor or a single tissue, those copy number changes are present in all of your cells in your body. So we typically expect that that means that we'll actually see an integer copy ratio, because you have an integer copy number in all of your cells. So your overall copy ratio is reflective of that. But we also expect that the germline copy number variants they tend to be smaller than the somatic copy number variants, because if you have a variant that's in all of your cells, you might not be able to tolerate such a large changing copy number. Like, you just wouldn't be able to survive that. Okay, so we obviously have talked about how somatic CNVs are drivers of tumors, but germline CNVs are also very clinically relevant. So on the one hand, they can actually increase your baseline cancer risk and give rise to cancer later in life. But they're also very important in rare diseases. So these are screenshots of papers just from 1 year and one publisher. There were 10 papers on the role of copy number variants in various Mendelian rare diseases. So these are super important clinically. Okay, so we talked about biologically what is a copy number variant, why should you care about that? Now let's talk about how that looks when we perform short read sequencing. So here on top we have a sample genome, which is diploid, and the reference genome, which is haploid, which we'll compare to. And you can see that there are some copy number variants in the sample genome. So there's a heterozygous duplication in orange. There's a homozygous deletion of the red region. There's another heterozygous duplication. And then there's a heterozygous deletion of the gray region. Okay, so what happens when we actually do sequencing? Here is what it looks like when we shear the DNA in the sample into fragments, and then we sequence those fragments and align them back to the sample genome. And then when we align them back to the reference genome, we start to see that the coverage varies in the places where we saw copy number variants in the sample. So there's an increase in coverage where there were duplications. There's a complete lack of coverage over that red region where there was a homozygous deletion. And there's depleted coverage over the heterozygous deletion in gray. So we can plot that as a very nice coverage profile that corresponds to the copy number across the genomic position. But in reality, it is a lot messier than that. This is what the kind of actual read count data that you would get from this sample might look like. And actually, you can no longer just look at the read counts and tell immediately what are the copy number variants. So even though it's germline, even though we expect integer copy states, it's still not so simple. So in this plot, we have the genomic interval position on the x-axis, and then we have a single sample, and the read depth is encoded by the intensity of blue color. So we can see that there is a lot of variation in the read depth across the genome for this sample, but we can't really tell where that variation indicates a change in the underlying copy number and where it's just noise. But when we bring in many other samples, we can start to see patterns. So, caveat that you can't actually call copy number variants by looking at this plot, but with a lot of math and computing resources, we can start to find patterns. And maybe we find that some of those samples that the samples are on the y-axis. Some samples have more variation in copy number. Maybe those are just messy samples. Some genomic intervals have more variation in copy number than others. Maybe those are also just messy intervals. Or maybe some of those intervals are true recurrent CNV hotspots where copy number variation is common in the population. So with many samples altogether, we can start to learn those patterns. Okay, so I've already talked a little bit about some of these things. Even though we expect to find integer copy ratios in germline CNV calling, which in theory makes it a little bit easier than somatic CNV calling. On the flip side, we are working with copy number variants that tend to be a lot smaller. As I said, if you have a really huge copy number variant like you see in a tumor, you might hit some really important genes, and then maybe you just wouldn't actually be able to survive that. So we don't see as many of those really huge copy number variants in the germline, although we see them sometimes. They're quite rare. So because those events are smaller, that makes it a little bit more challenging. Also, we would really like to develop a method that works with both exomes and genomes. We still have a lot of whole exome data that we're working with, and we'd like to be able to call copy number variants from that data. But whole exomes, the read depth is very, very noisy. They use PCR in library prep, but they also are enriching for the exome, right? So the entire purpose of whole exome sequencing is to capture just the coding regions. So by definition, the coverage is going to be uneven in an exome. Also, whole exomes only cover about 2% of the genome. So it's quite unlikely that we'll actually sequence the breakpoints of these structural changes. So we don't have that information about the breakpoints. And that leaves us with read count as our most reliable signal for copy number variant detection that exists in both whole exomes and whole genomes. Okay, so this is a little bit more metaphorical. The phenomenon that we're interested in, our signal, is these copy number changes in your DNA. So biologically, do you have fewer or more copies than expected of this particular region? But what we can actually observe and measure is the read count across the genome. And in between that signal and observation, there's a lot that happens. There's the sample extraction, the library prep, the sequencing, the alignment, and all of those steps, those epiphenomena, create noise but, as we go from our signal to our observation. So bear with me for this particular metaphor. Our observation is like this yin-yang symbol. It contains both the signal that we're interested in, the copy number variants, in red, and the noise, which is colored in black. So we have to be able to separate the signal and the noise in order to identify our copy number variants. Okay, so how do we do that in GATK GCNV? I'm going to go through some of the assumptions that we make and parameters that we set that kind of form the basis of the model that we're going to use to detect copy number variation from read depth. Let me just find where I am in my notes. Okay, so the first one here says that read count is subject to Poisson noise. what does that mean? When we perform DNA sequencing, we're essentially randomly sampling from the pool of DNA fragments and sequencing a somewhat random sample of them. We don't get all of the fragments, we get some of them. So we can model that randomness with a probability distribution, and we use a Poisson distribution for that. Second, there are going to be some samples and some genomic intervals that are just messier than other intervals. They contain more noise, so we call that overdispersion. So an example of that might be there was an issue in the library prep for a certain sample, and that shows up in the read count being more messy than in other samples. Okay. Next, there are systematic biases in the read count that make it not directly only impacted by the copy number. And those biases are determined by just a handful of factors that we don't directly measure that are unobserved. So we call those latent factors. So some examples that are listed here are GC content. This has come up before, that can influence how well you're able to sequence a region. Accessibility of the chromatin. If the chromatin is more open, then you're more easily able to sequence that region. And mappability is another potential factor here. The model itself does not actually define, or rather predefine or name these factors. It just kind of learns the patterns of the data. So it doesn't require you to predefine all of your latent factors. Okay, and for each of those different bias factors, they can influence the read count in different samples and in different genomic intervals with different weights. And we can learn those weights by looking at the patterns of variation across many samples and many intervals at one time. So there's a visualization on the top right here of that concept where we have our latent bias factors such as GC content and accessibility, and they're pointing to different genomic intervals with different size arrows. So to illustrate that there are different weights for different intervals. Okay. Number 6 is really our fundamental assumption that read count is in some way proportional to the integer copy number. Otherwise, it wouldn't be useful to us at all. Number 7, this is a parameter that we can set in the model. We say that the typical size of a CNV is on the order of 10 kilobases. In reality, there is a huge range of sizes of CNVs, but we kind of pick a number in the middle so that we get decent resolution across the whole range. Okay. Number 8 is talking about those recurrent CNV loci where copy number variation is actually quite common in the population. So remember, those repetitive regions can give rise to common copy number variants. So on the bottom right hand figure, we see a common CNV locus where in multiple samples there are CNVs. There's a homozygous deletion, there's a heterozygous duplication in 2 samples. So there's a lot of variation in copy number at that common locus. But outside of those common recurrent CNV loci, copy number variants are quite rare. And we actually expect to only find about 1 rare coding CNV per exome. So a vast majority, somewhere around 90% of the genome in each individual, or across individuals actually, is going to be copy neutral. We're not going to find copy or common copy number variation in most of the genome. So that corresponds to this de novo CNV over here. We see a heterozygous deletion in one sample, but no copy number variation in the rest of the samples. So that's not in a common CNV region. I'm just gonna take my sweater off, so give me a minute here. Okay. So based on those assumptions, which really come from our biological understanding of copy number variants, that is kind of what informs the framework of our model. And then what does the actual GCNV pipeline look like based on that? We start with coverage collection. Then we perform genomic interval filtering, where we remove genomic regions that are unmappable, that are in segmental duplications, that have very extreme coverage. So we are removing intervals where we don't think we're going to be able to do a good job calling copy number variants from the read depth. We are left with a set of high quality intervals where we can be confident in our CNV calling. Okay, then we perform sample clustering. So the GCNV pipeline is not designed to scale to like 10,000 samples at once. So part of this is for scalability. If you have a very large cohort, you will need to break it up into smaller groups of samples in order to be able to computationally perform CNV calling. But also, your quality of CNV calls will be better if you group together samples that have similar characteristics. So this is both for scaling and for improvement of the quality of the output. Next, we determine the ploidy. So what is the, how many copies of each chromosome does each sample have? So in humans, we would normally expect all of the chromosomes to have copy number 2, but in this step, you might find aneuploidies where an entire chromosome has been, you might have 3 copies of an entire chromosome, for example, and this is also where we determine the copy number on X and Y. So GCNV can handle that, mixing males and females together, because once we do ploidy determination, that serves as our baseline expectation for copy number on that chromosome for that sample. And any deviation from that baseline is what we are going to call a copy number variant. Okay, and now we really get to the action. This is where we are going to denoise our read depth data by learning the patterns of noise and bias and copy number variation to estimate the underlying copy number state in each sample at each genomic position. And that's an iterative process until we converge on a stable estimate for copy number. And from that, we can do CNV calling. So I'm going to take a drink of water and then I'm going to dive in a lot deeper to what that model looks like under the hood. Okay, so the approach that we take is generative modeling. So the idea here is that if we can simulate data that looks like the data that we observed by describing the relationships between the underlying parameters that we're interested in and the observed data, then through that process, we can understand our data, and then we can work backwards from the observed data to estimate our underlying parameter of copy number that we're actually interested in finding out. So just reminding you that there is a lot that happens between the signal and observation, and that's what we're going to try to model and understand. Okay, so the first underlying parameter that we look at is genomic interval class. So some areas of the genome are active. So those are those common CNV hotspots where we see a lot of copy number variation in the population. And a lot of the genome is silent, where copy number variation is quite rare. And the genomic interval class influences our copy number matrix of copy number for each genomic interval in each sample. Because as we can see here, we're much more likely to see variation in copy number in an active interval compared to a silent interval. So there's a relationship between those two underlying parameters. And both of those end up influencing our observed read count across samples and across intervals. And once we've described that relationship going in the forward way, we can then go backwards. We can use our observed read counts to estimate the underlying parameter of copy number. Okay, so the next couple of slides are going to have a lot of math on them, and I'm not going to talk about all of the equations. I'm going to try to just describe what are the concepts that are important for you to understand. But if you are someone who's interested in all of that math, I invite you to read the GCNV paper. There'll be a screenshot of that on a later slide that you can check out. Okay, so the first thing that we're modeling here is that genomic interval class, and we use a hidden Markov model, and it is sticky. And what does that mean? It means that if an interval is active, then it's more likely for its neighbor to also be active. So there's some probability that's related to the genomic position. Okay, and our copy number matrix, as we said before, depends on the genomic interval class. So we can see on the bottom right, in a silent interval, if our baseline expected copy number for that particular chromosome in that particular sample is 2, then the prior probability of having copy number 2 for that sample at that interval is much higher than the probability of having any other copy number. So basically, you're much more likely to be copy neutral than have any sort of CNV in one of these silent regions. By contrast, if we look at the plot for the active intervals, we see that it's actually pretty similarly likely to have any of these copy number states because copy number variability is quite common in these active intervals. In addition to depending on the genomic interval class, the copy number state is also sticky. So that means that if you have an interval that has a copy number variant, then its neighbor is also more likely to have a copy number variant, but they would actually be part of the same event. Okay, so now we've kind of mathematically described the relationship between our two underlying parameters of genomic interval class and copy number state. And now we want to describe the relationship between copy number state and our observed read count data. Okay, so this is a graphical representation of the factors that underlie the read count. So you can see that if you follow all of the arrows, all of them point to the read count because that's our observation, and everything else is just a factor that influences the read count. And we can write a bunch of equations to describe the relationships between these parameters. So first, we said earlier that the read count is a random variable. So we can model it with a Poisson gamma distribution, also known as negative binomial. Then we can write an equation to describe the relationship between the read count and all these other factors of the copy number state, the sequencing depth, mapping error, and our latent bias factors. We can write an equation that says that read count over dispersion, which is just extra noise that we weren't able to model with other variables, varies depending on the genomic interval and the particular sample. And our read count bias is a combination of both known and unknown latent factors, which have different weights in different individuals and different genomic intervals. Okay, almost done with the math. So now we have used this generative framework to describe the relationship between our underlying parameters, so genomic interval class, copy number state, all of these latent bias factors, and our observed data, which is that read count matrix. And it turns out once we have described those relationships with this generative model, we can then use Bayesian inference to go in the reverse direction and use the actual observed data to estimate the copy number state in each individual at each interval. And importantly, when we're using Bayesian inference, we don't just get a point estimate for the copy number. We're actually estimating the posterior distribution there. So we don't just get one number for copy number, we also get the probability that that number is the right one. So we get a confidence, which we can turn into a quality score for our copy number variant calls. And that's kind of all I'm going to say about the methods. But again, this is the GCNV paper, which was published earlier this year in Nature Genetics. So definitely check that out for more information about all of this. Okay, so now we've talked about copy number variants, and we've talked about the GCNV method. You know, what is it? What does the model look like under the hood? But actually, how do you then run the GCNV pipeline on your own data? So we start by pre-processing our intervals. This is probably going to sound familiar from somatic CNV calling. So our input intervals could be the entire genome, and we want to pre-process them into bins that are the right size for GCNV to operate on. Then we run collect read counts on each of our BAM files, and that will give us our read count matrix that shows us the read depth at every interval for every sample. Then we go back to our intervals list. We annotate it with the GC content, mappability, and whether that region is a segmental duplication. And we use those annotations as well as our coverage matrix to filter intervals. So we filter out the ones that are very repetitive or that have extreme coverage where we don't think we're going to be able to produce high quality CNV calls. And we end up with a filtered list of intervals where we're confident we can perform CNV calling well. Then we use those intervals and our read count matrix, and we determine germline contiguity. So again, this is where we're trying to find out how many copies of each chromosome each sample has, and that will become our baseline expectation for copy number for that sample on that chromosome, and any deviations from that baseline are what we're going to call CNVs. And here's where we actually train and run the model in a tool called Germline CNV Caller. So Germline CNV Caller is where we run, where we train that generative model and perform Bayesian inference to estimate the copy number state. And you'll notice that it's actually parallelized. So we don't run it across the entire genome all at once. So that's partially to help with scaling, so that we don't need like such a big machine, and it will also reduce the runtime. So instead, we run Germline CNV Caller across all of the samples, but across only some of the genomic intervals. And then once we have trained our model and estimated copy number, so we've kind of filled in our copy number matrix, then we run post-process germline CNV calls to turn those copy number estimates into actual copy number variant calls with quality scores and put those into per sample VCFs. Something I haven't mentioned yet is that GCNV actually can be run in two modes. So what we were just talking about is called cohort mode, where the pipeline is run and trained on a group of samples. But actually, there's another mode called case mode, where we can run GCNV on just one sample at a time. And this is because we have output the trained model parameters when we ran Germline CNV Caller. So we can take those trained model parameters and apply them on a brand new sample to perform CNV calling on that new sample, as long as that new sample was relatively similar to the samples where we originally trained our model. And that is called case mode. And it's a little bit simpler. So let's just run through it. We still need to collect read counts in our new sample. And we still need to determine germline contiguity. But then we can use the pre-filtered intervals and the pre-trained GCNV model to estimate copy number in our new sample, and then produce CNV calls for our new sample. And there are a lot of tools to run here. So they've been put into a scripted pipeline using WDL so that it's easy and automated to run this. And we can take advantage of the parallelization that you can do with WDL and Cromwell for scalability in both the cohort mode and the case mode. Okay, so you run GCNV. What do you get out of it? We will actually do a demo of this tomorrow, but I'll just tell you on a high level here what you can expect. So out of Germline CNV Caller, which is where we have trained the model, you get some sort of per sample output. What is the copy number at various intervals in that sample? You get the trained model parameters, and you also get some output that summarizes the loss function history over time during model training. So if there was, an issue with your output, you could go back and look at that loss function and see if you can find the issue there. Okay, and what is the output of post-process germline CNV calls? It is a single sample VCF with copy number variant calls. Each CNV call has a few different quality metrics. We typically recommend using QS as your main quality metric for filtering, but I want to also mention QSS and QSE, which are quality scores for the start and end of the CNV interval. So that's because, especially with exome data that is lower resolution, we might be very confident that there's some sort of copy number variant, but we might be a little bit less confident that we got the start or the end position correct. So the number of CNV calls that you should expect to get from GCNV without doing any filtering is about 10 rare CNVs per exome. It's obviously higher in genomes. But the precision, so that has very high recall, very high sensitivity, but the precision is a little bit lower. So we actually do recommend performing filtering if you are doing this for an application that requires high precision, such as maybe a GWAS. And after filtering, we expect to see more like one rare CNV call per exome. So let's say you ran GCNV and you're seeing way too many CNV calls in one of your samples, like over 200. What does that mean? That probably means that something went wrong, and the most likely explanation is that that sample was an outlier sample that was not similar enough to the samples that you used to train the GCNV model. So it had different patterns of variation in the read count, and GCNV was then not able to accurately determine where the copy number variants are. So if that happens, you should not use that GCNV output that has over 200 calls in an exome, but you could try retraining your GCNV model with more samples that are similar to that outlier sample. Okay, so I mentioned if you require higher precision, we recommend filtering. We've added some of our recommended filtering thresholds here. I'm not going to go through them. I've also been referring to the counts of rare CNVs per exome. So why is that? GCNV is actually aimed at finding rare copy number variants in exomes because the whole exome data, the read count is really kind of too messy to accurately call common CNVs. And so GCNV is not tuned to be able to do that. So in an exome, it's just really hard to distinguish between a noisy interval and a common CNV interval in exomes. But genomes are just less noisy as a data type. So it is possible to use GCNV to call common CNVs in genomes. And we do that as part of GATK SV, which I'll talk about tomorrow. Okay, so I mentioned that one step in the GCNV pipeline is sample clustering. So if you have a very large and heterogeneous cohort, we recommend that you split it up into multiple different groups of samples. This enables you to actually scale to that many samples. And also, you will get better CNV calls if you're training and applying your model on samples that have very similar characteristics, so that you don't end up with those outlier samples that have more than 200 CNV calls. So we have a method that we recommend in order to do this: collect read count over a set of intervals, perform PCA, and then cluster on top of that PCA to partition your samples into clusters. And then for each cluster, run GCNB in cohort mode to train the model on about 100 or 200 samples from that cluster. And then you can run case mode on the rest of the samples in that cluster. So why not just run cohort mode on all of the samples? It's because cohort mode is a little bit more expensive because you just have to do more when you're running cohort mode. You have to also train the model. It's still pretty cost effective. So it costs a little under 4 cents per sample to run GCNB in cohort mode in Google Cloud, and a little over 2 cents per sample to run GCNB in case mode in Google Cloud. And both of them take somewhere around 8 or 9 hours of wall time, but that's, remember, very highly parallelized. So under the hood, there's a lot more compute than just 8 or 9 hours happening. Finally, the performance and accuracy of GATK GCNB for detecting rare CNVs from whole exomes has really been heavily benchmarked. That's part of that GCNB paper that's in Nature Genetics. So this is showing results from an evaluation on about 8,000 exomes. They included quartet families so that we could find information about Mendelian violation rates. And they all had matched whole genome sequencing and microarrays to be able to compare to those orthogonal sources of data. And after high precision filtering, this data was benchmarked at about 86% recall and 90% precision, which is really good for CNV calling on whole exomes. And you can see in these top plots that both the recall and precision improve as the number of exons increases. So we can be more confident and do a better job in CNV calling from exomes for larger CNVs. We also see on the bottom the recall and precision compared to other exome CNV callers. So you can see that GCNV is like tied for best with for recall with a couple of other tools, XHMM and exome depth. But for precision, GCNV is way better than all the other tools. So that's all I have on GCNV. Any questions? Yes.
You have mentioned about the well-matched sample, 100 of the well-matched sample. How to define the well-matched, the trio, parent of the trio is the good sample?
Yeah, so you actually don't need families to make a well-matched set of samples for GCNV. You can use the clustering approach that I described later on in the slides to identify well-matched samples. So taking in read count over a set of intervals and then performing PCA and then clustering. So that will identify groups of samples that are well-matched. So really the sequencing method and the capture and things like that are what's going to influence how well matched the samples are, rather than like family structure.
One more question. Because in the hospital, they always collect the patient. Could we use patient as well-recognized, well-matched samples?
They don't need to be controls if that's what you're asking. You could get a whole group of case samples or patient samples and run them through GCNB together as long as they were sequenced the same way.
Okay, thank you.
I can ask a question about that. Don't you not want to treat parents? Can't want to know. For rare things, it's rare.
They'd still be rare enough. Yeah, so you can process families together. You just don't need to.
I have two questions. The first question is what is the recommended coverage depth of sequencing data, and the recommended sample size for effectively run GCNV. So that's my first question. My second question is, for example, my reference genome is like non-human draft genome, and can the copy number variant of the draft genome is important for GCNV to identifying the state of all the copy number variants in all the samples when I use the reference sample to map it.
Okay. Let me address your first questions first, except that I forgot them. Can you repeat that?
The recommended sample size and the coverage depth of the sequencing grids.
Okay, so I'm going to turn around so I can project. GCNV in cohort mode only requires like 100 samples in order to effectively train the model. So we recommend somewhere around 100 to 200 samples for cohort mode to train your model, and then you can apply that on as many well-matched samples as you want in case mode. In terms of sequencing depth for whole genomes, we typically use around 30x for whole exomes. I actually don't. Around 80x for whole exomes. At least that's what we commonly do in humans at the Broad. I actually don't know off the top of my head if they've like tested the impact of different depths.
Because you use the depth count, the reads count for calculating the CNV, right? So if just X5, how can you distinguish between like the copy numbers?
Yeah, we would expect that you need a high enough depth in order to get good results for sure.
Yeah. So my second question is is the CNV state of the reference genome is important to identify the CNV state of the samples that map to that reference genome.
So if there are like duplicated regions in the reference, how would that impact it? Yeah, so GCNV does end up filtering out some intervals that are part of segmental duplications because those can be really messy in terms of read depth because you will get reads, they might map to one part of the seg dupe, but they actually came from the other. So it's very difficult to do CNV calling from read depth in those segmental duplication regions. So, but that is something that GCNV could, that's why we have the filter intervals step of GCNV to remove those intervals where we don't think we can do a good job. So if you have annotations of where those seg dupes or other types of copy number changes in the reference are located, then you can pass those into the filter intervals tool, and it can take in that information and determine which intervals it's going to actually use for CNV calling.
So for example, if I have a read that map to multiple location of the genome, so should I remove it from the analysis or should I keep it?
You can, you don't have to remove it from your BAM file before starting GCNV, but you will want to provide those annotations of where those regions occur in the reference genome so that the filter intervals step can remove them if it doesn't think it's going to be able to do a good job.
Okay. Yeah. So thank you very much for your
Yeah.
answers.
Thanks. All right. Other questions? Maybe I'll go back to the front briefly until there's another question. All right. If that is all, then that was our last session for today. So thank you, everyone. And as usual, the instructors will stick around for a little bit longer for further questions. All right. Thanks.